across Pakistan. They'll be with us for the next 2 months, and our USDA cotton germplasm group is hosting them while they're here. So if anyone would like to set up a meeting with them, please let one of us know. And they also have their own objectives while they're here, and we'll also be contacting some of you to discuss various topics. Thank you.
Thank you very much.
Did you guys want to say anything, or?
Oh, thank you.
Just real briefly, this is Schuyler Smith. He's working on his master's here. He joined us from Iowa State, and he's going to show you that in a slide. But Schuyler recently got some preliminary data. He's going to talk about that, and then we have Rupa, who's another student in the program, in the corn program after that. And the rest of our guys are out riding a planter right now. So, Schuyler, please.
All right. Like this preliminary data, so I labeled this part 1 because it's just an introduction, and part 2 will be in the future, maybe this summer, maybe next semester. But so a little background, because the first time I present anything to you guys. I graduated out of state in 2012 with a bachelor's in genetics. While I was there, I interned for USDA soybean genomics lab with Michelle Graham, and then at DuPont Pioneer up in Wilmer, Minnesota with Steve Zalma and Lori Kerrigan, and at Monsanto and Huxley with Tom Carlson and Brian Gardenia, who graduated here, and then started in 2013 here. So my 3 projects for my master's. My first one was the blue corn, trying to find causal genes for blue corn seed. Another was the NIR oleic acid calibration, so trying to use the NIR spectroscopy to calibrate to be able to determine oleic acid levels, and then molecular analysis of the lines we have in our program, which I'll talk more about extensively later. So for the blue corn project, we have this open pollinated population called the Hillsborough white and blue, and so there's just this open pollinated population, so there's no selection done on it, and it's segregating for blue and white. And so someone took 4 years, so we had the OP 1, 2, and 3, and 4, and with those 4 years they took white seed and blue seed, planned them out, and saw that in the on the ears that were from the seed, all the white ones were all white and the blue ones segregated. So we have the homozygous blue and the segregating ones. And so it was pretty clear that the blue gene was qualitative. Whether it was 1 or 2 genes, I'm not really sure. I was trying to figure that out. And so from there we took 1 seed and planted it out, the 1 blue seed, from a heterozygous ear, and planted it out and got a heterozygous ear, and did the same thing through 2. So they were mostly isogenic siblings that I was using. So down here at this level, I took white seed and blue seed because they were related enough, except that the white they were segregating. So to try to determine, using high throughput sequencing, try to determine what gene was causing that, use a QTL study. So I planted out 15 different sibling combinations with white and blue seed, and I extracted all the DNA, had really good extraction levels, and sent it off to the lab to get sequenced, and they somehow lost all my DNA. So I haven't had time to get back to doing that yet. So that's as far as this project's gotten. The oleic acid project, there's the NIR machine if you haven't ever seen it. So I just took the sample, whole kernel, ground kernel, did both in here, it scans it. I get a readout on the computer. And so the population I was using is the QMS pop, which you've probably seen lots of this slide quite a bit. So it's a 4-parent meiotic syndesis population. It has 4 parents used at the top, and they're crossed, and then cross the end. So it's a 4-way cross. You have each genome lining up in syndesis at some point, and it'll self down a bunch. So you have all sorts of recombination and segregation going on in this population. And so it's good to get an array of the quantification of oleic acid. And so I did 3 rounds of sampling. I did 2 samples from Wesco 12 planting, and I took 20 originally. Those 20 are here, and so you can see the array of the segregation of color. That's kind of cool. I did 20 in the first, 45 in the second, and 39 in the last ones. I have 104 total. The software I've been using is called Omnic. So in the calibration, I have 80 samples now, and 24 are used for check, so of the 104. So I have the 80 that are used in the calibration to correlate with the NIR readings, and 24 to check to see how accurate that correlation is. And that's in TQ Analyst 8 software. For my third project, this is not my figure. This is Paul Nelson's figure in his paper called The Molecular Characters and Basic Inbreds with Expired U.S. Plant Variety Protection. So he took XPVP lines and lots of common public lines and did a SNP chip of like 680, I think, SNPs somewhere in that area, and did a principal component analysis and cluster analysis to see how they turn down. This is what his 3D image looks like. So you have like your O17, your B73A2 main heterotic groups, and you have OH43, and he has the cross here. You can see how they line. So our idea was to do the same thing with our lines to get prove how, because ours is mostly not from that same Midwest temperate zone, so we have a lot of tropical and subtropic corn. And so I got in-kind support with Elliot Hefner at Duke Pioneer up in Dallas Center, Iowa. And I got 274 of our lines were sequenced on 741 markers. And so this is what my data looked like after I spent months getting it to look this pretty. So now I used R and a distance matrix. I used power marker for the distance matrix. I used R to get my principal component analysis. And so this is what it looks like. In my first principal component, I have 10% of the variation, and in the second, I have 4.5 extra percent of the variation explained. So that was pretty good. And then when I plotted out, it looked like this. So that was fairly promising. I used structure to get the cluster analysis. And so I ran it through, at first I ran it through 15 levels of K, and that was more than I needed. I did this is how it defines the groups, and you have the red group and the green group, which I now know are the stiff stock and non-stiff stock groups. So the two main heterotic groups. And then at K equals 4, it's, I know now that it's separated out. The yellow group is a nonsense group that I'll talk about later, and the blue group is the llama group, our tropical, was Bavarian groups segregating out. Bolivia, that's what it is. But I did a posterior probability at the log likelihood that structure gives me. I have 3 different ways of measuring it here. So what I'm looking for is a plateau or a large peak in the data, like what happens here at 4. I have it here at 4. I wanted to see it around 3. And I think 4 is because of that nonsense group that I showed you earlier. I think if I deal with that group, it'll be more clear where my log, where my posterior probability says this is to find my actual level of K, where it should be. This is to find the best suited level of K to fit my data. And so it says it's around 3 or 4. I want it to be around 3, ideally, but this says 4, because it plateaus around there. This is my graph again. This is it with the cluster analysis added in. And so you have the stiff stocks and non-stiff stock splitting here. So that says that the first principal component is able to distinguish the difference in the variability between stiff stocks and non-stiff stocks, so that's pretty good. The third group is the nonsense group. You have all these red guys in here that they don't seem to make any sense. So I spent quite a little bit of time now trying to figure out what they are. And structure gave me that this guy here, here, and here, they're the 3 main ringleaders of that group, so they define a group and everything else is just associated to them. And so I've been trying to figure out, looking at the data that I showed you, the spreadsheet, what they have that's different. I think my biggest clue right now is that they have more heterozygosity than most of the other groups, and a lot of missing data too. So I've been trying to work around, see how I can either fix how they're interpreted by structure and power marker, or I don't want to eliminate them, but I might end up having to do that. So what K equals 4 for structure, this is where the llamas come out down here. So you have the non-stiff stocks, and the llamas are in that group, and they're down here. And then this slide is an 8, which doesn't really tell us much. But there's a slide that Seth showed 2 weeks ago, whoever's here had seen it, where we had the main inbred lines, the B73 and the TX775 or 2. I forget which one is the important one. We have a lot of TX77 things. But you have the main non-stiff stocks and the llama groups down here. And then the other thing that Seth had noticed that is interesting is that the second principal component seems to split the B104 group. So everything with a triangle here has B104 in it, and so it appears that the second principal component is splitting based on whether that is present in the pedigree or not. So that's kind of interesting. In my future, I want to finish the calibration and then scan. I want to see if I can do whole kernel instead of ground kernel. If you ever ground seed, it's kind of tedious, messy, and wild. So if I can do whole kernel, that'd be nice. I'd be able to get the whole population done, get a nice array quickly. I want to further investigate the structure analysis, figure out why that group is really acting weird, and see if I can get to assimilate into some of the other groups. And then redo my DNA extraction, because that'd be nice to have that. So I want to thank my Helly Hefner, who helped me at Pioneer, my committee members and advisors, Seth Murray, Russell Jessup, and Joseph Wika, and then Paul Nelson, got his paper I showed, and Jim Holland was also on that paper, and then everyone in our group. And this would be a great time for any of these. John?
So what's unique about the B104 line that causes it to have its own principal component?
Other than the fact that it's used in a lot of things, I don't know if it has anything that's unique to it. It's just crossed with a lot of things in our program. I would have to do a lot more molecular analysis to figure that out.
Yeah, I think it's just overrepresented. Might be the bigger issue when you look at all that stuff. And most of it isn't being used that much in the program. But he used a set of material that, as of 3 years ago, was sort of our core set of I think there were like a little over 300 lines in total, and then those sort of got whittled down.
Yeah.
And now we're down to 30, so there were a lot of B104s that sort of faded into the...
The other thing is the SNP chip that was used is designed for Midwest temperate material, and so with ours, it doesn't always predict very well. So it's designed to separate the stiff stalks and non-stiff stalks like it did, but then after that, it's kind of up in the air what it's doing in our material.
So is your molecular analysis, how is it going to help the breeders? I mean, stiff, non-stiff are two things by just visually we see them, and then this also shows the same thing. So how do we benefit from this molecular analysis as a breeder, maize breeder?
Right. So it helps prove novelty in our program, that our stuff isn't like anything else that's out there. You can see it on the graph. It will identify if there's a misclassification.
And you found that a few times.
Yeah, it's happened a little bit. And any contamination will show up in here.
And I also don't know if that's really true. I mean, you can't just go out in the field and say that's a stiff stalk, that's a not a stiff stalk. A lot of our material has sort of been crossed with tropical material, and so it's not really clear where it falls. And you can't make all the test crosses. It's just too laborious. So.
But looking at the diversity, molecular diversity between a set of inbred lines, can anyone predict the heterotic group, whether they belong to different heterotic group, or you still have to make physically cross and test it? That is the thing I'm looking for. If we can analyze group of inbred lines and predict these inbred lines will make a better heterotic group, that will be helpful. Is there any possibility of that?
Now that whole genome sequencing.
Anyway, hard for.
That'll be not easier work.
Good. Good.
Thanks.
Group of-